Hi everyone, it's Miss V. Happy Friday! Now, today we are going to be covering our final topic in the blood analysis unit. This means that next week we'll likely dedicate a few days to review and a small quiz or test before we move on to our next topic of anthropology. However, we're getting ahead of ourselves right now. Let's kind of rewind a minute because we still have to get through this topic. Now, we've covered a few things in blood analysis, right? The field of blood stain pattern analysis specifically requires your knowledge of biology, right? We had to learn how to detect different blood types and see what blood looks like under a microscope. You also have been getting comfortable with quite a bit of math and physics, especially, the Pythagorean theorem, sine, cos, tan, triangles, angles, all that good stuff to help you to measure the angle of impact as well as the area of origin. And now we're going to be talking a little bit about chemistry today. Now, we're not going to get too in-depth with it. However, chemistry is also an important field used in blood stain pattern analysis. And you might be wondering, what? Well, this picture down here will give you a very good hint. It helps you in detecting blood. Say at a crime scene where you think there's been a struggle, however, there's no blood. Maybe at the crime scene, you can now use different chemicals to test for blood compounds. And even if there is a stain, you may want to just double check that it is indeed human blood. So these different tests can help to confirm that yes, it is a blood stain. It is not just ketchup that someone accidentally squirted on the table. So we're going to go ahead and talk a little bit more about these tests and when you might use them and why. Okay. So the very first one we're going to talk about is when you arrive at a crime scene and you don't see any blood. Now, you may think, wow, there's signs of a struggle, maybe there's also a weapon that you find, but you don't see any blood. There is a test that some crime scene investigators will use to see if blood was indeed present and was cleaned up, or maybe it dried and kind of flaked off a little bit. Okay? Even with the most thorough cleaning, blood leaves residue. That is very difficult to remove. Okay? Now, you may have seen an image like this in a CSI TV show where they spray some magical chemical and all of a sudden, when they turn out the lights, everything's glowing that was touching the blood. That is not just a fancy CSI trick. It does happen in real life. It's a little less theatrical. However, it does happen. Now, what they're using here is luminol powder. And when you mix it with hydrogen peroxide, it is able to detect hemoglobin left behind by blood. Now, hemoglobin is a protein in your blood that's used to carry oxygen, and a lot of different chemicals that are used in blood detection are trying to detect hemoglobin, just like this one. Now, if blood is present, it actually turns this bright blue-green color that you see over here. And the really fun part is that it luminesces, meaning it gives off light for about 30 seconds. So it's best to do in a dark room. Really shows you the exact trail that blood has taken. Now, luminol reacts best with old blood, not really with new. So it's always best to try and do this with either old blood spatter patterns or ones that have tried to be cleaned up. Now, as you can see over here, what's really nice about it is that it can show you that specific spatter pattern or the trail mark, really helping to give a story to what happened at that crime scene. Unfortunately, on the other hand, luminol destroys the blood and it cannot be tested later on. So for further, blood typing and things like that, it's kind of been altered with the chemical, and so now you can't use it for different testing. So there's some pros and some cons to this method. However, you are still getting some valuable information that you may not have received otherwise if the blood was still hidden. So this creates a pattern that can help with the investigation. Another con about luminol is that it can actually accidentally also detect other substances like watermelon, radish, horseradish, and metals and paints, specifically copper. So there are some times when it can give you a false positive. Now, this is just testing for blood in general. It will test positive if it comes in contact with hemoglobin. However, it will come into contact with any type of blood and confirm. So this blood here in the bathtub, it could be fish blood, it could be human blood, it could be any animal of blood. As long as it has that hemoglobin molecule in it, it is going to test positive. I don't know, maybe there was also a watermelon explosion, right? So this is what we call a presumptive test. We are presuming that there is blood at the crime scene. However, we cannot confirm if it's human blood, but it's leading us in the right direction. So that's why it's called a presumptive test. So here's another image of what luminol looks like just on a carpet, and it glowing very brightly. And I'm actually going to show you a short little video or a little demo right now of what this actually looks like in real life.
All right, so I wanted to show you what luminol does in real life. So here I have a mixture of luminol reagent. So that has my mixture ready. And this here on my plate is just some ground beef blood from my dinner that I saved. Now I am wearing gloves because I am dealing with blood. Whenever this is ever something that you may be coming into contact with, it's always important that you're wearing this proper safety gear. And we're just going to go ahead and spray some of my luminol onto my plate, and hopefully we should get a reaction. Alright, so it is pretty faint on the video, however you can see that I have some definite glowing going on. This is actually the same chemical reaction that happens in glow sticks and in fireflies. Of course, they don't use blood, however, they do use a similar compound that gets the luminol to glow.
All right. So now we're going to move on to when you go into a crime scene and you do see the blood, okay? You see it plain as day. Maybe it's on the wall, a table, the ground. How can you test it to make sure that it actually is blood? Remember, it could be a Kool-Aid stain on the carpet that has been there for multiple years. Maybe it's not blood. Maybe it was ketchup on the wall, or my niece decided to do some splatter paint with red paint. We have to make sure that it is indeed blood. So how are we going to do that if we can see it? If blood stains or drops are found, we need to confirm that it's indeed blood, and you can do this with a Kastle-Meyer test. Now, this is a mixture of hydrogen peroxide again, but a different chemical called phenolphthalein. Okay? Now, the oxidation of hemoglobin that happens with the hydrogen peroxide in the blood produces a deep pink color. That's what the phenolphthalein does. It makes this really bright pink color. Right here, you can see it over here. This would be a negative test, and this would be a positive test for blood. So the swab will turn bright pink when blood is detected at the crime scene. And the really cool thing about this test is that it is very sensitive. With 1 drop of blood being present in a bucket of 10,000 drops of water, the test will still turn pink. So it's very, very sensitive. In fact, when I was trying this experiment out, I had accidentally gotten some phenolphthalein on my gloves, and suddenly my countertop was turning pink in spaces that I didn't even think I had, used the fake blood on. But it can show you just how small of a trace amount you need in order to turn that test positive. Now, the Kastle-Meyer test has the same reaction with human blood as it does with other hemoglobin-based blood as well. So that means that if it's horse, pig, fish, frog, whatever, human, as long as it is blood and it contains hemoglobin, it is going to turn pink. So it cannot guarantee that this is human blood, making it also a preliminary or presumptive test. So we will, in fact, know that it is blood. However, we do not know what animal it came from.
All right. So now we're going to try and test our phenolphthalein, or also known as our Kastle-Meyer test. So all I have here is a plate with some blood stains on it, and once again, these are just blood stains from some ground beef that I defrosted. I'm still wearing my gloves to make sure that I'm safe. And we're just going to pick up some of this blood stain with a cotton swab. Now, it might not look like I have anything on my Q-tip just yet. However, we're going to run this test because remember, we only need a very small amount for the test to become positive. So, very first thing I'm going to do is I'm going to add a drop of my hydrogen peroxide. We're just going to soak it in there. And then I'm going to add a drop of my phenolphthalein. Now, if it turns bright pink, that is positive for blood. And if it stays plain and white, that means that it is negative. So once again, you can see just how little of a blood sample you need in order to do this test. I didn't even affect the stain. However, this test also runs the risk of you altering the integrity of the stain. So once again, we have to weigh our pros and cons about doing these tests to make sure that all of our evidence from our crime scene is kept intact and is going to be able to be used in court.
Now, you might be thinking, okay, well, so these tests tell me if I have blood, but it's not telling me if it's human blood. That's where there's some more steps. So if blood is present, we need to be able to confirm that it actually came from a human. And you can do this by looking at blood microscopically sometimes. Now, all mammals, except camels and llamas, but it's okay, have circular, unnucleated red blood cells. So they're going to be tiny little circles, and they're not going to have anything on them. Now, a nucleus is like a really dark dot, like you see in some of these pictures. If it is a mammal, it will not have that dot. So, right, humans are mammals. So we have hair, mammary glands, things like that. That's what makes it a mammal. And so you will notice that their blood is also characteristically different from, let's say, other animals that are not mammals, like birds, fish, etc. They have oval blood cells with a nucleus. So right away, if we see anything like this over here, my bird blood has an oval and that nucleus in it, it's not possible for it to be human blood. Same with over here, fish blood also has that oval shape and nucleus. Frog blood as well as snake blood. Now these four options here are all very circular. We have horse, dog, cat, and human, and unnucleated. So it brings us a step forward, right? If our blood cells are circular and not nucleated, or they do not contain a nucleus, then we know it's at least a mammal, but we still cannot confirm that it's a human. So there's one other test we can run in order to ensure this. And it's the ELISA test. So this test detects and measures antibodies in your blood, right? Those things that are swimming around and trying to, latch on to antigens. Now, it reacts to human blood to tell if mammal blood is specifically from a human. So this is a much more specific test that is unique to human blood that can tell you whether or not blood came from a human. In some rare cases, it can be confused with chimpanzee or gorilla blood because it is very similar. However, in most cases, and hopefully at most crime scenes, you won't have to worry about gorilla and chimpanzee blood. This is a more confirmatory test. So I've talked about two different types of tests in this video. We have presumptive tests and confirmatory tests. Main thing that you should know about this is that a presumptive test can establish the possibility that a specific body tissue or fluid is present, right? If we use a Kastle-Meyer test or luminol, we know that blood is present. We just don't know if it's human blood. Whereas a confirmatory test will tell us specifically that a certain biological material is present, like human blood, okay? And that would be the ELISA test. Okay, so presumptive narrows down and helps decide which test to do next, whereas confirmatory is a very conclusive and final result, and it's usually going to give you a fairly definite answer. All right, everybody. Now we do have a little comparison chart that I'd like you to fill in, and have a wonderful weekend.